This video covers pyruvate oxidation and helps meet course objectives 3 and 4. As a refresher, glycolysis is a metabolic pathway that takes place in the cytosol under anaerobic conditions. Recall that a pathway may be anaerobic if it uses an electron acceptor other than oxygen. So, glycolysis is an anaerobic pathway that can occur even though oxygen is present but not used for this particular pathway. At the end of glycolysis, notable products include 2 molecules of pyruvate, 2 NADH coenzymes, 2 free hydrogen ions, and 2 net molecules of ATP. Most of the remaining potential energy from the original glucose molecule remains in the 2 pyruvate molecules. Under conditions where oxygen is available, pyruvate is transported from the cytosol through the porine proteins to the intermembrane space. Here it couples with protons and is transported into the mitochondrial matrix by a protein called pyruvate translocase. Pyruvate is then further oxidized in the mitochondrial matrix during a step otherwise called the linking or intermediate step. The enzyme complex called pyruvate dehydrogenase will carry out 3 important steps. It will decarboxylate pyruvate and release this as carbon dioxide, then couple it with coenzyme A to form acetyl-CoA and reduce NAD+ to form NADH and proton. Each pyruvate is oxidized by pyruvate dehydrogenase so that a total of 2 acetyl-CoA, 2 NADH, 2 protons, and 2 carbon dioxide are released. These reactions are simplified in this image. To the left, in the orange, is pyruvate in the cytosol. Once it's transported into the matrix, pyruvate dehydrogenase, shown as PDH, will decarboxylate pyruvate to release CO2, release NADH and proton, and join it with coenzyme A to produce acetyl-CoA. Also, note in this image the total number of carbon atoms in each molecule. Pyruvate enters as a 3-carbon molecule. With the removal of CO2, acetyl-CoA is a 2-carbon molecule. Also note that pyruvate oxidation does not produce ATP and is an anaerobic pathway. Once acetyl-CoA is produced, each molecule can drive a turn of the tricarboxylic acid cycle. In summary, both pyruvate molecules are transported into the mitochondrial matrix. Pyruvate is oxidized to acetyl-CoA and no ATP is made, but carbon dioxide is released along with NADH and hydrogen ion.